Welcome to today's webinar on Cannabis Inflorescence quality considerations, including stability data for EU-GMP Offtake. Essentially, what we're looking at today is what are the requirements of quality around cannabis flower when it relates to the export for medical grade. So I always like to start at the end in mind, which is to look at what are the quality considerations around flower in the international market. Why is medical cannabis different? What do people mean when they're trying to allude to a term like GMP flower, or what's the inference that they're trying to make with that term? Looking at requirements around good agricultural and collection practices, good manufacturing practices, the harmonization of methods around PICS and ICH, COA or Certificate of Analysis considerations around medical grade flower, and also looking at testing requirements for ISO 17025. Important to know is what is international offtake currently looking for to meet those agreements, and also which countries are open to international offtake. And I'd like to highlight why stability data is also important in this sphere. So today's talk has really been inspired by two publications. The first being from the Journal of Natural Products on the topic of cannabis inflorescence for medical purposes using the US Pharmacopeia considerations for quality attributes. If you haven't read this article as yet, I highly encourage you to download it, read it. It's extremely useful, and it's been followed up by additional guidance from the FDA on cannabis and cannabis-derived compounds, quality considerations for clinical research. And after reviewing these articles, I'd really like to just get into some of the material and review it. So, for me, how is cannabis flower currently sold in Europe? Generally, the Bedrocan model, which is the main supplier to the Office of Medical Cannabis in the Netherlands, they form a good model in terms of what could be called loosely GMP flower or inflorescence. Here is a good example of a COA where the cannabis floss of the Bedrocan variety, and I think they have 5 or 6 products of varietals, is sold into the pharmaceutical market, has an approximate concentration of THC and CBD defined. Its dosage form is flowers. It has a batch number. In addition, it has a grower specification. It has an irradiation date and dose. It has packager descriptions as well as testing laboratories, where you can see this batch was tested in September. And then it also defines the monograph that the Netherlands use in terms of defining their COAs. If you would like to get access to that monograph, feel free to call me, email me. I'll happily send that along to you. So let's have a look at what's in the COA. So the first few tests are really guided by the monograph, which relate to appearance, identity via microscopy or TLC testing, assessing foreign material and fineness, where obviously there's specifications and a conformance report to meet that specification. The micro side will now tie back more to European Pharmacopeia, where we would see a situation where specifically microbial assessment is done on non-sterile preparations, where we'd look at total aerobic microbial count, as well as total yeast and mold count, and gram-negative bacteria. And this is all outlined in the European Pharmacopeia. In addition, there's guidance on the absence of aflatoxins, where this is found in 2.8.18, and the absence of pesticides. I will say this about the COA from Bedrocan: there are limitations that relate to pesticides, specifically GCMS methods, but these are the tolerable and accepted limits within this COA, where you can see certain colony forming units or CFUs that relate to the microbe and molar concentrations. Right. Additionally, when it comes to the heavy metals, there's guidance within the monograph where this will typically relate to an ICP OES method and limitation. And there's some additional methods and monographs that are defined here, with the exceptions on the pesticides outlined. So the Bedrocan model is really one that's been working in Europe for a while through the Office of Medical Cannabis. There is a Dutch and German monograph available. They supply most of the flower within, obviously, the Netherlands, and they've been exporting to Germany and other European regions for quite some time. For the European regions that are importing from Canada, they tend to follow what would be a USP method, and I'd like to outline that and review that in comparison with the European Pharmacopeia. So, how is and why is medical grade different? Essentially, cannabis is cannabis. The medical grade is different in the sense that there's really a need for consistency. So, as much as there may be haters or lovers of the approach to medical cannabis, it really comes down to the need for consistency. If you're going to enter into a clinical trial for an investigational new drug application, you need to have consistent product every time. And Bedrocan has obviously been focused on trying to achieve this for their flower over years and years of stability or stabilizing their cultivation. In addition, I'd say consistency is key because if you're going to take this to a new drug application, you want to make sure that what you did in clinical trials is repeatable to a commercial scale. And this would effectively allow you for marketing authorizations in global territories. It has been done for flower. I would, however, say that it's a lot easier to achieve consistency and repeatability around concentrates and isolates because you have a more direct control over the actual percentage points within that framework. There's also a need for consistency on testing. I'm sure multiple people can relate to the fact that they have struggled with testing between labs, where you get one result from one lab, a different result from the other lab. You cannot have a situation where you test in the country of origin, get a result, and then you export that product for medical cannabis, and it fails testing on receival. And yeah, we really need to make sure that there's a stronger push towards ISO 17025 accredited testing labs, that there's comprehensive use of accurate standards, that those standards are stable, that they're not old or have exceeded shelf life, because ultimately your test is being compared potentially to some internal standards. And then also there's a harmonization around testing, that there's not too much of a variation that is happening in different regions of the world. Ultimately, all of this goals itself towards effectively establishing safety, efficacy, and quality of medical cannabis, but at a cost. So that's really the difference with medical grade and traditional cannabis. Now, I'd like you to take the following slide with a large pinch of salt, because it's something I just used internally, but I'd like to share it because what I've done here is I've just outlined the type of testing methods that are commonly seen across monographs and in different chapters, the typical instruments that may relate to it, as well as US Pharmacopeia and EU Pharmacopeia guidance that puts out chapters that relate to these testing methods. So let me use one as an example. It's on potency. Potency can be tested multiple ways: TLC, high performance TLC, NIR, or FTIR, but generally the accepted methodology would be the use of HPLC. I'm sure there are proponents of LC-MS, MS, but the dilution factors here may be inhibitory. So let's use HPLC as a workhorse, and then specifically which chapters within the USP relate to that method and the validation of that method. And again, pinch of salt, because there are going to be definitely additional chapters that relate to this, but I've tried to just outline at least something as a guidance post to be used when looking at this and to help drive standardization around the testing. In terms of the additional tests, terpenes, usually GC, headspace GC, your impurities for heavy metal, ICP-OES, pesticides. It's quite an interesting topic to discuss. It's also probably the biggest capital outlay for analytical testing lab, because it requires an LC-MS as well as GC-MS workflow that relates to that. And I also would like to highlight the importance of 561, which is Articles of Botanical Origins, as it relates to all of these tests for the most part. But definitely a topic for further discussion is pesticide testing, and I specifically would like to update this document in this slide and discuss with a colleague of mine who's had about 2 decades in analytical workflow to almost do a separate complete 1-hour webinar discussing the different methodologies and considerations that go into this. For now, I just wanted to outline this as a brief guidance post, and again, with a pinch of salt, but be aware that there are different methodologies. One thing that I found very interesting that's becoming more relevant is extractables that relate to packaging with specific guidance. Stability studies is something you will not see in a COA or certificate of analysis, but it's actually really important in terms of assuring the shelf life and the degradation that could occur with a product over a certain period of time, and I'll discuss that towards the end of this presentation and webinar. And then residual solvents really relate more to the extraction space and less so cannabis inflorescence. And these are some additional guidance chapters that you may want to look at, specifically in this space. Now, when it comes to analytical methods, you really need to look at the method validation and making sure that the validation of the compendial methods have followed the right process. Your harmonization is key across the different pharmacopoeias, but for me, method validation really looks to bring into frame your accuracy, your precision, your repeatability, your specificity, your detection limits, the quantification limits, the linearity and range within that. And again, this will be a completely separate discussion as we can go quite vertical and granular on these points. Main thing for me is the agreement on standardization. I like to use the analogy of monopoly because everyone wants to pass beginning and collect $200, and you want to make sure that when you send a product off for receipt that it passes testing and there are no surprises. So, highlights the points I've mentioned on the ISO accreditation, the need for consensus on methods. I feel you're completely within your right to understand what methodologies were used in terms of the testing, having a look at the possible chromatographs that the testing lab has done, understanding what instruments were used, making sure those instruments are within calibration, etc. Obviously, when it comes to validated methods, making sure those internal standards are accurate, have a good shelf life, are stable, stability of those standards, as mentioned. And then there's obviously a need for harmonization across the different testing and regions. I think the hottest debate in this space would probably relate to pesticides, where this is a bit tricky for a couple reasons and should be discussed more. But you know, whether that's the Canadian list or the Oregon or California list, or let's use an example, if you're sending products from South Africa to Israel or to Australia or to Germany, you know, what is the right pesticide testing list to use? Should it be the Canadian list? Surely not, because we're not in Canada. So getting some form of harmonization around pesticides and knowing specifically what your offtake is looking for in terms of the different testing methods is kind of critical to be successful. On regulatory framework, I'd like to bring up a few things. Good agriculture and collection practices is generally, you know, the starting point around cultivation, and I have a colleague I'll bring in for discussion on this point. On GMP, you know, there's been a big push, obviously, with all the conversion of the Canadian good production practices to GMP. GMP is thrown around a lot in discussions. I would just really like to highlight the need for distinction between food and pharma grade GMP. GMP for pharma is definitely more related to the CFRs for 210, 211 and important for medical cannabis for certain. A lot of GMP that's out there usually pertains to food or dietary GMP, so be on the lookout for that in the definition. Pharmaceutical Inspector Convention or Cooperation Scheme, so PICS. PICS is basically the attempt to harmonize the different GMPs across regions. So to give you an example, there are 54 PICS member states in the Southern Hemisphere: Argentina, South Africa, Australia, New Zealand, and in the Asian region as well, Japan, and then a lot in the North American region, Canada, North America, Mexico, and most of Western Europe. So yeah, PICS is looking to standardize GMP across regions. So if I know that I have a GMP manufacturing plant that is in Mexico or South Africa, that it should conform, and the inspectorate bodies for those regions should hold to the same standard for GMP as, for instance, France or Germany or Canada, as an example. So this constant push towards harmonization makes it a lot easier for international trade within certain countries, and this is going to be a big topic going forward. The other thing to look at is the ICH, the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, as they also have a big part to play in terms of harmonization. Now, countries often open to offtake agreements. We need to maybe discuss this. For me, targeting a country that has an existing medical program is key, because you'll know there will be a good absorption in that market. Good examples will be Australia, Israel, Germany. Countries that have reimbursements for medical cannabis, definitely going to be growing economies and spaces for medical cannabis. You need to also make sure there's no protectionist policies in place. I often cringe a little when I hear about, oh, I have an offtake that's set to go to Canada. I think it's been established for a while, and definitely in this publication in August, that Canada has been accused of protectionist policies on blocking of imports. And if you would like to have a look at this for yourself, you're welcome to go to MJ Biz Daily. I've included the link. But this is where it's important to have multiple offtake options and also to know the requirements for those different regions and to ensure that there is no protectionism around that, because the last thing you want to do is build your entire business model around an offtake to a certain region and then there's an implementation around protectionism. Currently there's no MRAs or mutually recognized agreements for cannabis in Europe. I did mention there's a Dutch and German monograph, but if this day ever eventually came where there was an MRA for Europe, definitely help in terms of cross-border movement of medical cannabis and the standardization, harmonization. Important to know the clarity on the COAs for testing and the requirements. You need to know what types of pesticide lists are going to be accepted within the region you would be exporting medical cannabis to. Prior stability data on previous batches, also important. So stability is usually done in hindsight. So, you'll do your COA and your testing, you'll send a product, but increasingly with multiple batches being done this way, you want to have started to establish stability data to just support how stable that product is. There has been recalls in the past with several companies due to the final product not meeting specification, and stability data will usually allude to this early on. Countries that are interesting in terms of offtake is obviously Israel overtook Germany in July of 2020 in terms of the major importer for medical cannabis, but this was also followed up shortly in September with a more stringent review of their imports and the origin countries and the GMP qualification around those imports if it related to extracts or oils, as well as the quality of the flower or inflorescence that was being brought in. So you definitely want to be on top of this to ensure that if there is a more stringent requirement from a regulator or port authority, that you are at the highest standard to ensure that there are no issues in terms of your supply chain and QMS or quality management system. Stability. What is stability testing? Essentially, if you need additional guidance, you can refer to ICH, topic Q1A, revision 2, which relates to stability testing of new drug substances and products or finished pharmaceutical products. The purpose of stability testing is to provide evidence on how the quality of a drug or substance, where medical cannabis is that, varies with time under the influence of temperature, humidity, and light. Essentially what you're doing is you would take a sample, you test that sample, and we can discuss how to sample effectively for samples, but take that sample, have a repeatable version of that sample, stick it into a stability cabinet. And if you run it in a stability cabinet for 6 months, it would be an accelerated stability test. If it's going to be the year-long stability, you would have some data. When you retrieve that sample from stability, you would test it again and see what the degree of degradation is. So it would ultimately inform you, what are the results from the stability testing and how does it advise on product degradation and recommended storage conditions and shelf life claims. And this is generally a requirement around products, and even as it relates to COVID vaccines, the one thing that will be rushed is the stability assessments. Generally, there are 4 climatic zones in stability, the primary ones being zone 1 and 2. Zone 1 in the northern regions of the hemisphere, and zone 2, South Africa, parts of South America, Australia, New Zealand, as well as the lower regions in North America, specifically the US. You can look at increasing the stringency or going for like a zone 4 or zone 3 stability testing, but it would seem that, especially for inflorescence or flower, that the more extreme you go in your stability, the more likely you're going to have unwanted or undesirable results from that stability testing. So just make sure you know which zones you're going to be moving cannabis to when you do stability, and that you are performing those stability tests in conformance with those zones. You're welcome to always send an email to ask more and discuss it further. I've done some previous discussions on extraction, so feel free to look out for that if you found this chat informative. and like to get some information on that. And as I said, this is just a brief summary of a topic that I found really important, which was cannabis inflorescence quality considerations and how that relates to international trade of medical cannabis. Feel free to always ask me questions on email, to call, and as I said, if you're looking to get hold of the Dutch monograph specifically for cannabis testing on inflorescence, happy to send that to you. You send me an email. I hope you found this informative and useful, and please be on the lookout for upcoming discussions in this space. Thank you for your time.